Hello, and welcome to your Microbiology Bio 203 lecture video on microbial requirements for growth. I am Mr. Kennedy, and I'll be sharing with you the factors or conditions under which microbes grow best and eventually how we evaluate that growth. First thing we're going to do is take a look at the physical requirements and chemical requirements for microbial growth. Bacteria, microorganisms are a lot like people in that we all have specific temperature, pH, osmotic pressure, and even nutrient requirements that we prefer in order to grow. So it shouldn't surprise you that microbes have specific temperature ranges, pH ranges, and osmotic conditions under which they grow best. It should also not surprise you that there are nutrient requirements or chemical requirements that microorganisms require in order to achieve maximum growth. And even aerobic conditions, oxygen, carbon dioxide levels, can affect this growth. As we examine growth and we consider the rate at which an organism grows, the first thing we're going to look at is temperature. Temperature for a microorganism can be measured in the form of minimal growth temperature, optimum growth temperature, and maximum growth temperature. Each of these hopefully is fairly self-explanatory, but when you consider temperature ranges that microbes prefer, they again are very similar to large organisms such as ourselves. There are minimum temperatures that we can tolerate. There are maximum temperatures we can tolerate, and then there are, well, the most ideal conditions that we prefer. So if you're living someplace that gets really, really hot in the summer and you choose to continue to live there, well, then maybe that hot summer temperature is the maximum growth temperature that, you can tolerate. If the temperature exceeds that maximum temperature, then you're probably going to consider moving. And the opposite is true for the minimum temperature. At the optimum temperature, you're happy no matter what. In terms of temperature, we can sort organisms based on the temperatures they prefer into some specific categories, which are named here for you. I prefer to talk to you about them in this graphic form. So here you can see that we have, 5 different categories to sort organisms by the temperatures that they prefer. So you take something like a thermophile, which really, really likes temperatures between 40 and about 72 degrees Celsius. The 40 degrees Celsius measurement would be its minimum growth temperature. The 60 to 62 degree temperature range would be its optimum, and then the maximum would be at about 72 as it experiences temperatures that are exceedingly warm. Just to kind of give you some specifics, like here's the sacchartrophs, which grow best between 0 and 20 to 30 degrees Celsius, which are important for food spoilage. We take this information about temperature growth and we can apply it to our daily lives in a lot of different ways. So here's a couple of thermometers, and we can use the knowledge of the temperature range that a microorganism prefers for growth to either promote or inhibit its growth. One of the reasons why we cook things is to kill bacteria to protect us from pathogenic organisms. So by elevating a food item, let's say, to temperatures above the maximum growth temperature tolerable by a microbe, then you can effectively eradicate that from your food, at least for a short time. While your food sits out on a counter and starts to cool off, it will be recolonized by microorganisms that are in the air. And depending on how long it sits out and what temperature it sits at, growth of microbes can return. So we need to limit the amount of time that we leave food items exposed to the air in what is referred to in this picture as the danger zone of temperatures, approximately 15 degrees Celsius up to about 50 degrees Celsius. That's why we refrigerate things. Once things, food items have been used by us, if there's leftovers, we should immediately take them and put them in the refrigerator, driving their temperature below 15 degrees Celsius to prevent further microbial colonization. Okay. So, just to kind of give you, some more terminology associated with temperature, we also have our thermophiles. The thermophiles have an optimum growth temperature of 50 to 60 degrees Celsius, and they're found in hot springs and organic compost. Hyperthermophiles, optimum growth temperature is above 80 degrees Celsius. This is just a picture of those places. Next, pH. pH, again, like most living things, is a measurement of acids and bases, and we all have a set pH that we prefer. Most bacteria grow best between a pH of 6.5 and 7.5, while molds and yeasts grow between 5 and 6. Acidophiles really like acidic environments. Now, osmotic pressure. Osmotic pressure is a measure of the solution's osmolality compared to the cells that are in it. So when you think of osmotic pressure, I want you to think of the terms hypertonic, hypotonic, and isotonic. Hypertonic environments have more solute in them than inside the cell. So that basically means if I put something in a hypertonic solution, that the cell will undergo what's called plasmolysis and basically have all of the liquid literally sucked out of the cell. Okay. This obviously would be fatal for the cell, shriveling it up like a little prune. If I put something in a hypotonic environment, then the opposite scenario may play out. There will be less solute outside of the cell than inside, and fluids would rush into the cell and could cause the cell to lyse or explode. Okay. Extreme or obligate halophiles actually require high osmotic pressure or basically high salt concentrations in order for them to survive, while facultative halophiles tolerate high osmotic pressure, but they don't necessarily need it to survive. This is just a picture illustrating a plasmolyzed cell versus a cell in an isotonic solution. In an isotonic solution, you'll notice the cell looks pretty normal. In the plasmolyzed cell, all of the liquid is being sucked out of the cell. The cell basically prunes up inside of its cell wall and would perish. Okay, we've actually been taking advantage of this scenario for literally generations in food preservation, using salt to cure things or even to smoke things like meat to preserve it long term. By smoking or salting things, you basically pull the water out, and then any bacteria, microorganism that might land on it later on can no longer grow and or survive because the water literally gets sucked right out of it. Okay. Chemical requirements. Next thing to look at are the chemical requirements, and I like to consider this like the nutrients that cells need in order to survive. We'll start with carbon. Carbon is a structural backbone of all organic molecules. Chemoheterotrophs use it as an energy source, and autotrophs use carbon dioxide not as an energy source, but as a building block for food for photosynthesis. Nitrogen. Nitrogen is a component of DNA and ATP. Most bacteria will decompose protein material for their nitrogen source, but some actually use atmospheric nitrogen in the form of nitrogen gas. Some will actually use ammonia or nitrates from other organic material, which could be like a waste product from an animal or some sort of liquid from the decomposition process going on as another living thing is broken down. Sulfur. Sulfur is used in amino acids, thymine and biotin. Most bacteria decompose protein for their sulfur source. Some bacteria will use sulfates and or hydrogen sulfide, in their metabolic processes. Phosphorus is used in DNA, RNA, and ATP, and it is found in all of our cell membranes in the form of a phospholipid. Phosphates are the source of phosphorus for microbes. There are a few trace elements that are required for life. These are inorganic elements required in small amounts. They can usually be used as enzyme cofactors and include things like iron, copper, molybdenum, and zinc. When you think about the trace elements and cofactors, there are things that we call cofactors and coenzymes. If the item in question is inorganic, like iron or copper, it's a cofactor. If it is organic, then it would be considered a coenzyme. Oxygen. The oxygen requirements for a microbe are very, very important to controlling its growth. Obligate aerobes always require oxygen. They have to have oxygen in order to survive. They are just like you or I. Facultative anaerobes can grow via fermentation or anaerobic respiration with no oxygen, but they probably would prefer to be in an oxygen environment if you asked them. Obligate anaerobes are unable to use oxygen and are actually harmed by it. So if you want to kill an obligate anaerobe, literally all you have to do is expose it to air. Aerotolerant anaerobes can tolerate but cannot use oxygen, and microaerophiles require oxygen concentrations lower than what is actually present in our air currently. This is just an illustration from your textbook to illustrate the growth that we would see from each of the different oxygen growth scenarios. So if something was an obligate aerobe, then their growth would be concentrated at the top of a test tube, anaerobe at the bottom, etc. Some other things to consider when considering oxygen is a form of oxygen referred to as singlet oxygen. Singlet oxygen is oxygen that is boosted to a higher energy state, and it is reactive. We get singlet oxygen anytime oxygen gas is split from its O2 form into individual oxygen atoms. Each individual oxygen atom is reactive, and there's lots of different ways that we can deal with it and that it shows up in cells. Superoxide radicals occur when we take oxygen in a state where it is carrying extra electrons and overall negative charge and combine it with hydrogen. When doing so, we make hydrogen peroxide and oxygen gas. Peroxide anion is going to occur when oxygen is carrying two extra electrons. And again, we take hydrogen peroxide in this scenario, and we can split it into water and oxygen. The next thing we're going to look at are organic growth factors. As I mentioned a moment ago, the inorganic growth factors, or cofactors, are things like iron and copper. The organic growth factors are things like vitamins, amino acids, purines, and pyrimidines. From here, we're going to shift gears a little bit, and we're going to look at biofilms. Biofilms are microbial communities that are formed by masses of microbial cells that form slime or hydrogels that adhere to surfaces. Bacteria can communicate via quorum sensing, share nutrients, and shelter one another from harmful environmental conditions. They can almost function in a manner similar to a multicellular organism, although they are still individuals. Biofilms can be found in the digestive system and sewage treatment systems. They can clog pipes, and they are 1,000 times more resistant to microbiocides and involved in 70% of infections of catheters, heart valves, contact lenses, and even cavities, dental caries. From here, we'll look next at culture media. Culture media is the material that we're going to grow our microbes on. Culture medium contains nutrients prepared for microbial growth. If it is sterile, there are no living microbes in it. The organisms that we put on the growth medium is referred to as our inoculum. Once growing, we refer to that growing group of microbes on our culture medium as a culture. The material that we choose more often than not for a culture medium is agar. Agar is a complex polysaccharide used as a solidifying agent for a culture media in a petri dish, plates, slants, and deeps. It's generally not metabolized by microbes, liquefies at 100 degrees Celsius, and solidifies at about 40 degrees Celsius. If the culture media is chemically defined, we know its exact chemical composition. Fastidious organisms are those that require many growth factors to be provided in a chemically defined medium. So in order to have a chemically defined medium, you need to know the exact material that is put in the medium, down to the gram. Complex media is made up of extracts and digests. Things like beef broth would be a complex medium. There's no distinct down to the gram measurement of exactly what you put in the medium other than it was beef broth. So when you use a term like nutrient agar or nutrient broth, you're using complex medium. This is a table just to kind of give you some ideas of what might go into a chemically defined medium for growing a chemoheterotroph such as E. coli. And you can see that we know the exact gram by gram constitution of the medium. This is a defined culture medium. And again, in a defined culture medium, you know exactly what's going into it on a gram by gram basis. This is the composition of nutrient agar. Nutrient agar contains peptone, beef extract, sodium chloride, agar, and water. you'll notice that since it has beef extract in it, then we would consider it a complex medium. Next are reducing media. Reducing media are used for the cultivation of anaerobic bacteria. They contain chemicals like sodium thioglycolate that combine with oxygen to deplete it, or we might use heat to drive off the oxygen. This is an example of a anaerobic growth jar where we'll actually use envelopes containing carbonate, activated carbon, ascorbic acid, and water to produce carbon dioxide and create an anaerobic environment for our microbes. Anaerobic environments can also be maintained through hoods such as this. Capnophiles. Capnophiles are microbes that require really high carbon dioxide conditions. In those cases, we'll have to use a CO2 packet as just illustrated, or a candle in the container to produce CO2 to support growth of these individuals. In special culture techniques, biosafety levels always have to be maintained. It's important that you know what you're dealing with and work within those biosafety levels at all times. Selective and differential media. Selective media suppress unwanted microbes and encourage the desired microbes. They'll often contain inhibitors to suppress growth. Differential media allow distinguishing of colonies of different microbes on the same plate. Some media have both selective and differential characteristics. This is a picture of blood agar, which is a differential media containing red blood cells. Here you can see the bacterial colonies literally digesting the blood through hemolysis. This is also a differential medium, and in this case we have Staphylococcus aureus and Staphylococcus epidermis plated on a plate very similar to a mannitol soy agar plate. In mannitol soy agar plates, some bacteria can actually digest the mannitol and make the plate literally change color from, as you see in this case, a red color to a yellow. Enrichment cultures. Enrichment cultures encourage the growth of a desired microbe by increasing very small numbers of desired organisms to detectable levels. They're usually done in liquid form. To summarize, this is our culture medium and definitions of each type I mentioned before. Chemically defined, the growth of chemoautotrophs and photoautotrophs and microbiological assays, requires chemically defined culture media. Complex media can be used for the growth of most chemoheterotrophic organisms. Reducing media is only for obligate anaerobes. Selective media is used to suppress unwanted microbes and encourage desired microbes. Differential media differentiates colonies of desired microbes from others, and an enrichment culture or an enrichment media would be similar to a selective media, but designed to increase numbers of desired microbes to detectable levels. Pure cultures. Next section we're going to look at is how do we obtain a pure culture. A pure culture contains only one species or strain. A colony basically is a population of cells arising from a single colony, and ideally in pure culture, that colony would be the relatives or lineage of a single variety. A colony is often called a colony-forming unit and can be separated from a larger group of organisms using the aforementioned media or a streak plate method to isolate the pure culture. This is how streak plating is done, and you can see that you start off with a microbial lawn that eventually differentiates out into individual colonies as you drag your loop through the inoculum and spread it out into thinner and thinner layers on your plate. Next thing we're going to look at is preserving bacterial colonies. To preserve bacterial colonies, you have several options. Deep freezing can preserve a bacterial colony by reducing its temperature to -50 to -95 degrees Celsius. Lyophilization is similar to deep freezing, but it's done in a vacuum, at which point all of the water is driven off of the microbes and they are basically stored in powder form. The main difference between deep freezing and lyophilization is that vacuum environment. In deep freezing, the organisms would have to be kept cold until you're ready to use them. In lyophilization, they are freeze-dried. So once that freeze-drying process has occurred, then you can let them warm back up and they will not grow until water is added. Next, we'll look at bacterial division, or this process of growth through reproduction. Bacterial division increases the number of cells, not the size of the cell, through activities like binary fission and budding. Binary fission are the two primary asexual forms of reproduction that I'd like you to focus on. Binary fission is very similar to mitosis. The cell simply divides in two in a manner nearly identical to mitosis without using mitosis terminology, because mitosis is the division of the nucleus in a eukaryotic cell, and these cells are not eukaryotic, nor do they have a nucleus. Therefore, we simply refer to it as binary fission. Budding, as its name implies, involves the cell simply pinching off a copy of itself, and in either of these cases, the end product is the production of a cell that is a genetic clone to what you started with. This is a picture of binary fission, and you can see that a single cell's genetic material is duplicated, and the cell simply splits itself in two without using the mitosis phraseology, but still many of the same similar steps. Generation time. Generation time is the time required for a cell to divide. Now, there's no set generation time that applies to all microorganisms. As a kind of a standard, most bacteria have a generation time of anywhere from 20 minutes to 24 hours. Binary fission will double the number of cells in each generation. So to calculate generation time, you take the total number of cells and basically multiply that times 2 raised to the number of generations that have occurred. So if you started off with 5 cells and those 5 cells went through 5 generations of reproduction, you're going to take 2, raise it to the 5th power, multiply that answer times 5, and that will tell you how many cells you're going to have after all of the division has been completed. To kind of get an idea of what it looks like visually, you can look at this table. Okay, so as you look at this table, you can see that you're going to start off with one cell that eventually is going to go through 5 generations of reproduction, and after 5 generations of reproduction, there will be 32 cells. Another way to look at that is illustrated thusly. At this point, again, we're stressing the idea that you have one cell, and it's going to go through as many as 20 generations, okay? And by the time it's all said and done, after 20 generations, you'll have upwards of 1 million cells. You will not have to worry about graphing this on tests. You just need to be able to do the calculations. Now, one graph that you do want to pay attention to is not the log graph for cell division, but instead this one that maps out lag, log, stationary, and death phase of growth. This graph illustrates points during the life cycle of a microbe where it starts off struggling to survive in the lag phase or reproducing just enough to offset losses. At some point, the log phase kicks in, at which point you have a higher survivorship and the numbers increase exponentially. At the stationary phase, births and deaths, if you will, equal one another, and the graph levels off. During the death phase, you have more cells dying than are being made, and the population declines. Finally, we need to look at how we measure microbial growth. Direct measurements count microbial cells. Examples of direct measurements include plate count, filtration, MPN, and direct microscopic counts. In plate counts, we go and count colonies on the plates that have anywhere from 30 to 300 colony-forming units, or colonies. So we're not counting necessarily individual cells, but instead colonies of cells. To ensure the right number of colonies, the original inoculum must be diluted using serial dilution. Counts are performed on bacteria mixed onto a dish with agar using a pour plate method, or spread on the surface of the plate using a spread plate method. This is an example of serial dilution, taking small inoculums from one test tube, diluting as it goes, and then transferring that to a plate. This type of analysis is done frequently in food and drink preparations to determine the exact microbial load of items that we are ingesting and what is safe. This is an example of the pour plate method. where the material is poured onto the plate and agar is added to it on the left, and on the right, the spread plate where the material is added to the plate, and then we use a spreader to distribute it. Filtration. In filtration, the solution will be passed through a filter that collects the bacteria. The filter is transferred to a petri dish and grows as colonies on the surface. MPM, most probable number method. This is a method that involves the utilization of multiple tube tests and count the number of positive tubes that you get, and you compare with that to a statistical table. In the MPN, you will simply take a nutrient medium set of 5 tubes for a set, add your inoculum in different dilutions, and then look for the number of positive tubes in each set. You will need a table from a Burgess guide in order for you to then look at your confidence limits and determine the number of colony forming units that you have. Direct microscopic count is what its name implies. You take the volume of bacteria in suspension, place it on a slide, average the number of bacteria per viewing field is calculated, and then you use the special Petrov-Hausser cell counter and determine the number of microbes that you're looking at. This is what a direct microscopic count would look like. We can estimate bacterial numbers by indirect methods. Probably the easiest indirect method involves turbidity. Turbidity is the measurement of cloudiness with a spectrophotometer. Basically, if a solution is nearly translucent or see-through, then your microbial load is low, and vice versa. We can also use metabolic activity, which is the amount of metabolic product that is produced proportional to the number of bacteria, or dry weight, where the bacteria will be filtered, dried, and weighed using filters of some sort. Okay, that is the end of your notes on microbial growth.